All right, guys. So we are going to go ahead and get started today. And before we start, I do wanted you guys to remind all of you that April 15th, that is the next week, is going to be the plant lab practical through Blackboard. The same rules will follow. And do not forget, there are several things due that particular week, right? As usual, your last homework is due, and your lab notebook for the plant component, right, is also due. Um, and then also remember that the last activity of the entire lab is going to be on April 29th. So April 15th, we do not meet in the class. You will have the class time, the entire class time. I want you to use that for doing the practical, right? I will, as we always do, I will open up the lab practical about 15 minutes before the lab starts, and I will also keep it available for another 15 minutes after the regular lab time. So it's the same like last time we did. The same rules apply, right? And then on the 19th, that particular week, we do not have any labs. And the last week, that would be the week of April 29th, or on April 29th, we will have the poster presentations. Just want to make sure whether that date is correct, April 29th. Yeah, April 29th. So 15th lab, um, the week of April 19th, that is actually April 22nd, we don't have labs. Again, April 22nd, no labs. And April 29th is the poster presentation day. And poster presentation, we will meet in the lab as usual through the same Zoom link where you will be doing poster presentations. And let me also ask you to make sure that you know which part you are presenting, right? And also remember to upload your 2 peer reviews on the poster presentation date, right? Um, by the poster presentation time, your team, you can actually upload your presentation, the team evaluations, team assessments, right? And then after the presentation, you can also do the assessment on the rest of the presentation teams, right? Now, both of those peer assessments are required, and you will receive a grade for uploading that. Any questions regarding the next few weeks before I start what we are going to do today? All right, I don't hear any questions, so I'm going to move on to today's lab activity. All right, so we are going to talk about flowering plants today, right? And this is the phylogeny, and you do notice that actually they all are single lineage, a monophyletic lineage, right? A sister taxa to the gymnosperms, right? And the most notable feature is actually evolution of two related features, right, that are unique to that particular lineage. The anthophyta, or the flowering plant lineage, is the flower evolution, and then of course the flower after fertilization becomes a fruit. Okay, those are the unique features that I want you to be aware of. Again, do notice that they are also part of the seed plant, right? That's why they share a common ancestor with gymnosperms. Because of that, gymnosperms are a sister taxa to the angiosperms, and collectively all of them are seed plants, all of them are also vascular plants, and all of them are land plants. And their evolution sort of actually happened concurrently with the evolution of gymnosperms. And that is the only phylum that we have for angiosperms, right? The species richness is huge, right? Now according to current knowledge of botany, right, that we have, clearly flowering plants are much more diverse than any other land plant, right? Any other green plant. But the problem that I'm hesitant to say that out loud is that um, the bryophytes or the non-vascular plants as well as avascular plants are not that well studied. Secondarily, do not forget that extinctions have happened, on all vascular plants, avascular plants, and gymnosperms, right? So we don't know for sure like whether angiosperms are the most diverse. But if you look at the evolutionary history, right, there are lots of there's a greater degree of speciation happening in angiosperms, and angiosperms are well adapted to occupy different environments, to occupy different niches. So I wouldn't be surprised if angiosperms are truly diverse than any other land plant, right? But do know that we haven't completely studied all non-vascular plants and all mosses. Okay. Um, Angiosperms are almost completely woody plants. There are a few non-woody plants, right? But most of them are like overwhelming number of those are woody plants. They actually have a woody stem. Um, and what is a woody stem? Well, remember from the gymnosperm, those are the plants that have a secondary growth um, in the stem, and that secondary growth also happens in the root as well. Um, Now they do have all the kind of growth forms. That's why I'm saying almost completely woody, but not entirely woody plants. Yeah, lot of them are woody plants. There are some of them are climbers. Could be vines, could be lianas, and then some plants actually have a very short stem, like onions, right? They are called bulbs. Some of them are herbaceous, right? Where they grow up to a level of a shrub, or like maybe not even, to the level of grasses. Um, and so there are lots of habits or growth forms, right? Very tall rainforest trees to extremely small, tiny shrubs and herbs that you find in a grassland, or grasses themselves, or sedges. All of those are actually, are very short, stunted growth angiosperms. However, their life cycle, resembles the basic plant life cycle. The main differences between angiosperms and gymnosperms include double fertilization, right? Now, remember, when I introduced you to gymnosperms, I did mention about the probability of there is not probability that there are certain groups of angiosperms undergoing double fertilization where two sperms fertilize two eggs. But remember, in the case of gymnosperms, right, this only one of the eggs actually grow beyond the zygote level and become an embryo. The second fertilization in gymnosperms undergo abortion. Okay, so yeah, technically, if you consider overall picture, like having, if I were to mention two fertilization happening, yes, some gymnosperms undergo two events of fertilizations, right? But the second fertilization, not both zygotes develop. One of them aborts in gymnosperms. In angiosperms, um, the double fertilization continues. One becomes embryo, the other becomes the food storage, right? And the second characteristic is actually having a flower, I would say. The true flower, I did mention that some gymnosperm reproductive structures appear like a flower, but they are not true flowers because the seeds are literally naked, they are open, right? But in a flowering plant, in an angiosperm, the actual seed is completely protected or covered within is covered within a flower, right? So that's actually one major difference. The second or the third part or related difference is evolution of a fruit, right? The flower, upon being fertilized, become a fruit, right? The fourth characteristic is actually production of a flower. Yeah, I did mention the flower already. So those are the three characteristics. There's a flower, and the flower becomes a fruit after fertilization and true double fertilization, right? All of those are actually characteristic features that I expect you guys to know very well. What else do we have here? Yeah, those are actually the characteristics I want you to know. And then pollination, remember pollination was something that you learned in the angiosperm world, but remember that now in flowering plants, there is actually use of animal pollination. Earlier, the pollination was largely based on wind for gymnosperms, but now there are animal mediated, insect mediated, bird mediated, bat mediated. There are so many different animals that can help pollination, and that kind of very extravagant level of pollination, animal mediated pollination, is actually a angiosperm character. Now, Anthophyta is the only phylum that you find within angiosperms, and there are several groups of these anthophytes: monocots, dicots, right? Monocotyledons and dicotyledons, and there are many other groups like magnolids, eudicots, but for the sake of simplicity, we are only covering the monocots and dicots, during the lab. Your lecture might actually go a little bit broader. Now, this is a graph that I already shown to you when I covered other plant groups. I do want to highlight several features. Now, some of you got this part incorrect in your last homework, right? So let me explain that one more time for the sake of clarity, right? When you're looking at seed plants, right, the sporophyte is dominant. The gametophyte is reduced, smaller in size, smaller in terms of its longevity in the life cycle. And the gametophyte is dependent on the sporophyte for nourishment, right? So, but in the case of ferns and fern-like species, fern-like vascular plants, sporophyte and gametophyte are independent. Okay, now, and again, if you look at the seed plant gymnosperms, seed plant um, life cycle, right? The microscopic female gametophyte occupies inside the flowers, right? And then the microscopic male gametophyte, right, occupies also inside the flowers, right? So in other words, gametophytes, both form of gametophytes, there are two gametophytes, male and female, right? And both of them are microscopic. Both of them occur inside the flower, and the flower is located on the sporophyte. Okay, now that's extremely important to understand. And also, other cool things is that regardless of cladistics, the gametophytes are always haploid. Sporophytes, regardless of the species, are always diploid, right? This kind of a comparative picture is quite helpful for you to remember stuff, know stuff. Now, again, this is not a new picture. You have already seen this. This is regarding the characteristics or the derived traits, synapomorphic features of seed plants. All of them have smaller gametophytes, microscopic gametophytes, right? Heterospory is universal, microspores and megaspores, right? Presence of ovules, right? It's a new feature you only see in seed plants. Pollen, another new feature you only see in seed plants, right? And because of the pollen evolution, the water is not necessary for the sperm to swim. Now, do not misunderstand. Do they lose sperm in the seed plant world? No, right? The sperms are kept inside the pollen and released from the pollen into the female reproductive structures, right? Into the pollen tube, to be specific, right? So because of the pollen presence, the pollen will carry the sperm closer to the female reproductive structures, right? So don't say that they have lost their sperms or sperms become pollen. That's not the case, right? The sperm does not go anywhere else. And then, of course, evolution of the seed, right? It's also a seed plant characteristic, and where the seed comes from is actually, seed development happens within the female reproductive structures. Seeds are better equipped to survive in the external environment. They can be carried from the parental plant to elsewhere, right? They can actually become dormant for a very long time, can be transported across a long distance by the means of animals, by the means of wind, by the means of water, right? And grow elsewhere. And it actually carries the developing embryo as food supply to nourish the developing embryo. And it is protected by a seed coat. That kind of a structure you did not observe in the spores, right? Now, plant anatomy, like you do need to realize that in ferns and in all the vascular plants for that matter, including ferns, angiosperms, and gymnosperms, you observe through tissues. Now, this is, most of you got this part right. If you got it wrong, right, the difference between through plant tissues and other plant structures are actually presence of vascular tissues, right? Leaves, stems, roots, all of them have vascular tissue, right? Thalloids, thalluses, rhizoids do not have vascular tissue. Okay, that's the only difference. Now let's talk about the anatomy and the parts of the leaf. We did not cover that kind of parts in the previous lecture, right? So this particular part, the leaf has two components, right? Leaf has two components. This part, the larger, component, which is actually mostly responsible for photosynthesis, is called the lamina or the blade. I like the word blade better because it's more English, right? So think about it in actual blade, like, the thing that you have in your razor, right? It's like a blade. It's flat, it's expanded, right? And then petiole is sort of the stem of the leaf blade, right? That actually connects the leaf blade to the stem, to the actual plant stem, actual plant trunk, right? And then if you want to go a little crazier, there are more advanced labeling you can do, right? So this particular part of the leaf blade is called the leaf base, quite literally the base of the leaf. The other end, which are usually pointy, is called the tip, right? And in certain leaves, the tip is not pointy, it could be round. And the edge of the leaf margin is called margin, right? And then the venation of the leaf blade has two parts, well, three parts. The central part is called the midrib or the central rachis. And then the other parts that sort of radiates into the leaf blade are considered veins. And the veins can divide again into venules, right? Okay, moving on. Leaf can be basically of two types. Simple leaves, like in this case, where you have a one single undivided leaf blade, or can be compound, like in these cases, right? So compound leaf, in this case, in wood sorrel, you actually see a single leaf blade divided into three leaflets, right? We call them trifoliate because there are three leaflets. And in the case of Virginia creeper over here, you actually see a single blade divided into five leaflets, right? So there are two types of compound leaf. Palmate is actually when the leaflets are arranged like the palm, like fingers of your palm, right? And if the leaflets in the compound leaf are arranged similar to a palm frond or a palm leaf, right, where the leaflets are arranged side by side, right, you consider that as a pinnate compound leaf. And then, well, leaf arrangement can also be defined as how leaves are organized within the stem, right? Over here, you actually have alternate leaf where the petiole, right, are directed in opposite directions, right? Sort of having an alternate arrangement where in a single leaf node, right, we only see a single leaf, right? And then on the next node, you see another leaf where it is directed towards the opposite side. Now, and then we have the opposite leaf, right, where you actually have two leaves emerging from a single node over here. In the alternate leaves, right, leaves alternate up the stem. from one side to the other, right? In the opposite leaves, you actually have two leaves emerging from a single leaf node where both leaves are direct towards the opposite directions. Okay, now last one is this, the rolled leaves. It's where you actually see multiple leaves, 3 or more, right? Not 2, 3 or more, arising at the same leaf node, and those leaves are arranged like a flower, right? There are multiple leaves radially arranged around a given leaf node, around a particular node, right? So those are the three basic forms. Now, let me actually show you this particular diagram where I'm going to put a fourth form. Now, basal form is a modified version of the rolled version. So the only difference is you only have like a single set of leaves, right, arising from a single node, right, and the rest of the plant does not tend not to have any other leaves, right. So usually basal structures are very small. Those are very stunted in growth. They are very short. And then, well, a reminder of the stem anatomy, right? What is a leaf node? Well, you already know, right? You know, the leaves tend to arise from the leaf node or the stem node, right? And so leaves, where leaves are attached are where you find the node. The distance between two nodes are known as an internode, between nodes, internodes, right? And then you need to know about two kinds of buds. One is the terminal bud. So it's the growth of the terminal bud that actually elongates the stem, right? There are terminal buds on the main stem. There are terminal buds at the end of each branch. Now, what kind of the bud that you find at each node above the petiole is considered either a lateral bud or the auxiliary bud. The same thing, same term. Now, those lateral buds are responsible for producing new leaves or producing branches from the main stem or producing flowers. So an auxiliary bud can either produce A, a new leaf, B, a branch to the main stem, or produce a flower. Okay. And how trees grow laterally, right? It's expanding. How trees expand, how they grow, like, you know, literally speaking, fatter, is by growth of the lateral buds. How trees go taller is by the growth of the terminal buds. Now, yeah, that's actually basically what I want you to know in terms of stem anatomy. Now, here it's more diagrammatic feature, right? You know, with the same labeling. Stems could be of different types, right? Yeah, most of the stems that you might be aware of are above ground, right? They grow against gravity, anti-gravity, right? They grow tall, but not all the stems are like that, right? So the first kind of stems that you see over here, right, in this wood sorrel example, another example could be clover, is actually stolon. Stolons are actually modified elongated stems that run horizontally along the ground or sometimes immediately below the ground, right? Instead of growing vertically, these stems are growing horizontally. Um, and another type is rhizome, right? Rhizomes are also modified, elongated, horizontally growing stems that occur below ground. Now, rhizomes are deeper in the ground than stolons. And another kind is tuber, where actually again those are deep underground stems, where the stem is actually expanding, right, into storage organs. Rhizomes are also storage organs, but rhizomes are much more smaller compared to tubers in terms of their size, right? And we can eat both rhizomes and tubers, right? And then the comb. Now, do not mistake yourself. Combs, this you will think, oh, are they onions? No, right? They actually, the example is actually sword lily, right? They look like onions, but they are not even closely related to onions. Completely different families from onions. Onions are actually on my bottom image. Those are considered bulbs. Combs are actually short, erected, upright, erected, modified underground stems, not above ground, underground stems, um, And in that case, the leaves could be completely absent or very small, reduced to scale-like structures, and the short stem itself becomes swollen to store food, right? And bulbs are actually looks physically similar to the outside. They are very similar to combs, but bulbs actually have a very short disc-like, extremely reduced stem, right? Now, if you look at an onion, right, the bottom, right, which is actually hard and white where roots are coming off from, that tiny part, right, which is like actually a few millimeters in thickness, that is the actual stem in an onion, right? The bigger storage components of the onions are actually leaves, right, not the stem itself. So comb is actually um, a completely different from bulb despite their appearances. In fact, I do consider bulbs as a modified stem where modification includes being reduced in size and having absolutely very little to no function, right? Moving forward, again, another much more clearer picture of all the modified kind of stems. Since bulbs are rather modified leaves than a modified stem, I eliminated onions from an example, right? So the examples would be for stolon. I mean, I said clover earlier, right? And then for rhizomes, this is actually ginger is a good example, right? Which is actually an edible component. For tubers, potato, any kind of a yam is a good example for tubers. And then corn, I mentioned sword lily, right? The entire family of sword lilies could be considered examples for corms, where you can see the true stem is actually, getting fatter, swollen as a storage organ, right? Yep, that's actually it. And like I showed these examples, so since this is a true stem, buds can come out of it, right? If this is actually a bulb, you will not see auxiliary buds coming out of a bulb, right? From an onion, for example, right? And then you do in tubers, if you look at a potato, you do see this kind of tiny, weird structures coming out of it, like protrusions coming out of it. Those are auxiliary buds. They can actually develop, right, into other tubers or other independent plants when these things grow. Actually, if you plant a tuber, a potato, right, it will grow into a potato plant by development of the auxiliary bud. Same thing regarding rhizomes, right? Rhizomes over here is actually a auxiliary bud coming out of a rhizome, right? And then, of course, all of those structures have these roots. They're adventitious roots, and I will explain you what an adventitious root is. Adventitious roots basically are roots that help anchor the plant, that absorb nutrients and water from the substrate, right? And adventitious tissues can come out of anywhere from the plant, not necessarily from the root itself. It can come from any above ground plant, right? Can come out from the stem, can come out from branches, can come out from certain parts of the leaf, right? So adventitious roots can emerge from anywhere of the plant, not just from the below ground components. Okay. Now let's talk about these roots, right? Now this is a taproot, right? The central large part that actually grows deep into the soil is the taproot. In a carrot, the part you eat is actually a taproot that modified into a storage organ. Now, taproots are characteristic of any angiosperm, right? But if you compare that with a dicot, a monocot versus dicot, dicots retain the taproot, but in monocots, they first actually have a taproot, but as the monocot plant mature, the taproot disintegrates, and instead of a taproot, they actually have these fibrous roots, where from the stem, right, many independent roots arise, so those are called fiber-like roots, fibrous roots, right? Now, adventitious roots, right, are what you see over here. Like I mentioned, adventitious roots can emerge from the above ground, from the stem, anywhere in the stem, not just the base of the stem, anywhere. Can they come out of the node? Yes. Can they come out of between the nodes, internode? Yes, right? So the adventitious roots can randomly arise and grow into the substrate from anywhere of the plant, right? Again, a diagrammatic picture over here, right? More detailed picture of a dicot, right? So this is the taproot, which is also considered the primary root. Branches of the taproot are considered secondary. Secondary roots can further branch, making tertiary roots, right? And anywhere within the plant root, particularly towards the more younger part of the plant, right, you actually see structure called root hairs, which are miniature, extremely small, microscopically small roots, right? And primary roots, secondary roots, tertiary roots, their job is to anchor the plant, stabilize the plant, root it literally into a substrate. The parts that are more capable of absorbing water and nutrients are actually the root hair. So root hair are located, see, like on the the younger part or the developing part of the taproot or the primary root, as well as in equivalent parts of the secondary and tertiary roots. So you have taproots, you have hair roots in any younger developing parts of the root system. Now let's talk about the seed anatomy, right? Here you're looking at dicot seedling, right? The external covering is the seed coat, and then this larger, the two-halved part are actually the cotyledons. Cotyledons technically are the first leaves to emerge that also perform a storage function. Then you have this stem, embryonic stem, right? Then you have the actual embryonic root, also known as the radicle, and then root hair that are actually emerging from the embryonic root, right? And there are two parts of the stem, right? And I will actually explain that two parts of the stem later. Now let's look at a monocot seed, right? The same parts. You have the seed coat externally protecting the seed, a single cotyledon, not two halves of cotyledons, a single cotyledon, right? And then, of course, the radicle, the embryonic root, and then the hypocotyl, which is the embryonic stem, right? The larger part of the stem, I said stem has two parts, epicotyl and hypocotyl. Hypocotyl is sort of the more longer, more larger part, right? So I will mention about both of them in a subsequent picture. And another part is endosperm. Now you may ask the question, okay, you showed me an endosperm. What is the endosperm? Immediately you should notice that endosperm is triploid, has 3 sets of chromosomes. Everything else in the seed is diploid, 2 sets of chromosomes, right? Let's go back over here. I don't have an endosperm. If you look at over here, you do have an endosperm. Well, it's not that endosperm is absent in dicots. That is not the case. The thing is, endosperm is there. It's extremely small. It has become shrunk because the cotyledons of dicots takes over the role of the endosperm, right? Endosperm's job is to function as a storage organ, to feed the emerging embryo. What happens in the embryonic development in dicots? The cotyledons absorbs the nutrients from the endosperm, and endosperm starts to shrink, right? But in monocots, the endosperm remains as the nutrition source, even when the seedling continues to grow. Now, again, to dicot plant parts, the top above ground part is considered the shoot system, which has leaves, stems, and flowers, right? Of course, in this one, you don't have flowers. The buds, everything, right? Nodes, internodes, all of those are part of the shoot system. The opposite is the root system. Root system grows towards gravity. Shoot system generally grows against gravity, right? And then, of course, remember stolon, those are the parts of the shoot system that could be actually above ground or below ground, right? Immediately above ground or immediately below ground, right? So that's a dicot plant. In a monocot plant, you have the same system, the root system and a shoot system, right? And the difference is the root system actually has multiple fibrous roots rather than a taproot. They had a taproot as a seedling, but as they grow and mature, they lose the taproot and develop fibrous roots instead. Over here, you actually do see the taproots and the secondary and the tertiary roots emerging from the taproot. Flower anatomy, right? This is a very important part. It's extremely important you know this general parts that I'm going to go through, because then actually when we learn about fruits and seeds, you have to remember what I'm teaching over here. So what are the parts? Well, let's start from the bottom. This particular part, sort of the um the sort of petiole-like part of the flower, right, is called the receptacle, right? It sort of holds the rest of the plant parts together. So hence receptacle. Then these green parts, right, that are sort of located on the bottom of any flower is called a sepal. If given plant is going to have multiple sepals, right? And collectively, all sepals are known as the calyx. Above the calyx, you actually have the colorful components. Sort of a colorful, larger version of sepals are known as petals, right? Now, sepals could be photosynthetic. Petals usually are not photosynthetic because petals actually have different color pigments, not chlorophyll, right? Now, that colorful part, the petal part, there are multiple petals in a given flower. Collectively, all the petals are considered corolla, right? And I don't know whether that is any, like where the Japanese got the name for their car, Corolla. Don't know where that comes from, but in this case, it's actually all petals together is considered corolla. And then you have female reproductive parts and male reproductive parts. All the male reproductive parts are collectively called stamen. Stamen has two parts. The head of the stamen is considered anther, where you actually have pollen, and then this stalk of the stamen is called the filament, right? And look at the female reproductive part. It's called collectively the pistil, or the way I like to call it is actually the carpel, right? Now, are there differences between the word pistil and carpel? It's extremely agonizing, but for you guys, I'm completely okay with you using pistil and carpel interchangeably, right? But there are the female reproductive parts can become modified. So if I tell you that pistil is the same as carpel, you might think, oh, okay, that means actually this particular thing. The entire reproductive system of a flower is a carpel. So only a single carpel per flower? Well, not always. Sometimes what happens is this pistil could actually get divided themselves into multiple carpels, where you actually have sort of compartments within the pistil where each compartment is also considered a carpel. So deal with that very cautiously, right? There could be instances where you have multiple carpels within the pistil, right? And pistils, carpels, all of them are parts of the female reproductive system of a flower, right? So pistil has three parts: stigma, the head of the pistil, the stalk-like part, style, and the bottom rounder part, ovary. Ovary is a pretty important part because that's where you see the ovule. Ovule, as it matures upon being fertilized, becomes the seed. Right now, understanding these very basic parts are extremely, extremely important for your lecture and the lab. So if these parts are not clear, if you want me to go over, please holler, interrupt me and holler, right? Now fruits. Fruits are, as simply put, mature fertilized ovaries. Now, I have to always tell you the exceptions. Like you guys have realized by now, biology is not a black and white discipline. It's gray, right? So you can ask me, okay, wait, professor. So if a flower does not get pollinated or fertilized, you don't get fruit. There are certain instances where actually certain plants reproduce parthenogenetically without fertilization. So yes, in many cases, fruits can develop without being fertilized. And in agriculture, in crop farming, farmers could actually, induce fruit development by hormones, right? By providing, like by spraying hormones on the plants without fertilization. So yes, fruits can develop, flowers can become fruits without being fertilized. But in nature, for the most of the time, flower must undergo pollination to become a fruit. Okay. Um, Now, fruits are mature ovaries, after they get fertilized for the most of the cases. Vegetables, what's the difference between fruits and vegetables? Well, vegetables are any part of the plant that isn't the ovary, right? So stems can become a vegetable. Leaves can be considered as vegetable. Certain developed roots, developed flowers can become vegetables, right? I mean, for example, cauliflower is a vegetable. That's a flower becoming developed. Kale, that's leaf-based vegetable, right? So potato, root-based vegetable, right? And some things that we generally in the market, in the household, when could call it a fruit, they actually aren't fruit. Certain vegetables that call it a vegetable is actually not a vegetable, right? For example, tomatoes, it's a vegetable, right? But it ain't. It's actually a fruit because it's a mature ovary that actually becomes the tomato. Strawberry, it's actually a vegetable. It's not a fruit because what you eat, the stuff that is fleshy, right, is actually in the ovary. What you really eat is a modified version of, I will go, is a modified receptacle, right? So in fact, when you eat strawberry, next time take a look at the surface of the strawberry. You see this tiny, 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 seed-like things embedded on the external surface of the strawberry. That is actually not just the seed, that is the fruit. So when you eat a strawberry, are you actually eating the fruit? Well, when you eat a single strawberry, you are actually eating a lot of real fruits, right? So generally, what you eat, the part that you desire in the strawberry, is actually a vegetable part, right? But inadvertently, unintentionally, you actually eat the actual fruit as well. So it's pretty interesting and complicated. There are many different types of fruits. Two basic types, fleshy and dry. Fleshy fruits are literally fleshy. They are more moist. There's a lot of water content in the fleshy fruits, right? They are softer. Blueberries, orchards, oranges, cucumber. See, like cucumber, you consider that as a fruit, but it's not. Cucumbers you consider as a vegetable, but it's actually a fruit, right? A fleshy fruit. Um, and then there are dry fruits. There are two types of dry fruits based on how the fruit splits into seeds when they mature, right? Dehiscent, the first type, is actually when the actual fruit splits into different parts. Indehiscent is actually non-dehiscent, meaning they are not splitting apart at maturity, right? So most of the nuts that you eat, like hazelnut, and sunflower seeds, and many maple seeds, corn, right, ash seeds, all of those are indehiscent. They don't completely split apart at maturity, right? Dehiscent is actually when they completely split apart, right? Most of the legumes, for example, peanuts, for instance, is actually an example for dehiscence. Now, few examples for different types of fleshy fruits. I provided lots of types over here, but I'm only going for a handful of them. I believe in the lecture you might cover a few more, but I'm going to keep it simple here. Palm, right, is actually the apple is a good example for the palms, right? In that case, the ovary is surrounded by the fleshy hypanthium, right? Now, hypanthium is a new term. Hypanthium Okay, where was the hypanthium in a flower? Hypanthium is collectively when sepals, petals, and stamens, when all of these three parts join together, they actually make the hypanthium, right? And in apples, actually the hypanthium becomes fleshy, right? And you get a palm, right? So that's what, when you eat an apple, what you are eating, this part is actually the hypanthium, which is a overgrown, fleshy collection of sepals, petals, and stamens, right? Um, and the actual ovary is here, okay, right over here. Now, and then the next part over here, right, we are looking at tomato. So in tomato, it's actually a berry. You may never believe it. It's a berry, right? Um, and why is it called a berry? Well, berries are produced when the fleshy part is mostly produced out of the pericarp. The pericarp actually is the modified ovarian wall, right? Now, the ovarian wall, when it develops into a fruit, has three parts: endocarp, mesocarp, and exocarp. Exocarp is the external most covering. Endocarp, internal most covering. And meso, in between covering, right, is the mesocarp, right? So in fact, um, in the pericarp, it is the mesocarp that actually mostly become fleshy, right, in a tomato. Third group is actually Hesperidium, right? Example. That's also a berry type, in which case the exocarp actually becomes a tough, leathery covering. And actually the peel, the take off, scientifically we call it the rind, right? So that is actually, when you're taking off the rind, you are literally taking off the endocarp, okay? You're taking off the mesocarp. Oh, and sometimes actually when you take the peel off, you're taking endocarp, mesocarp, and exocarp, all of them out, right? You might leave parts of the endocarp around the actual fruit, right? So the example for Hesperidium is the lemon, right? So in that case, what do you mean by a Hesperidium? It's a berry type where the pericarp has modified into a leathery rind, a leathery peel, right? Okay, and then drupe is actually, you have a fleshy fruit, again, the pericarp, right? The mesocarp, to be specific, or parts of the pericarp has modified into a fleshy, softer structure. The actual seed, if you look into, right, is actually had a very hard endocarp, right? Very tough, thick endocarp, right? And the actual seed is located inside the endocarp, right? And next time you eat, um, oh my gosh, um, the next time you eat a peach, right? You can actually, what you are eating is the mesocarp, right? You are not eating the endocarp. You throw away the endocarp. Actually, if you cut open, if you like, like chew the endocarp, and you can actually see the seed inside, right? The last part is a peepo. Peepo is another berry, right? Which actually has a very hard, thick peel rind, right? That's why watermelon is so thick. And the mesocarp actually makes up the more fleshy edible component. What you eat with yellow in color or red in color, pink in color, that is the mesocarp. The rind, the thicker exocarp, you sort of throw away, right? So you learned today that the watermelon is actually a very unique kind of a fruit. It's a berry. Now, talking of dry fruits, right? First the indehiscent type, which actually retain their seeds and do not completely crack open after the seed becomes mature, right? There are several types I'm going through. Akene is where the seed is only attached to the seed coat at a very smaller point location. In contrast, grain is the opposite, where the seed is completely attached to the seed coat. The best example is corn. Samara is the third type, where actually it's a modified form of an akene where the seed actually has modified wings that help them to disperse through wind. Schizocarp is a situation where the entire fruit breaks open into like it doesn't completely crack open. When they actually breaks up partially, it actually divides into carpels, right? Remember what carpels are. Carpels are that entire reproductive unit in a female structure of the flower after it gets fertilized. After when the seed matures, you get the pistil part becomes a carpel, sometimes into multiple carpels. So in this case, right, each carpel has a single seed inside it, single mature seed inside it, right? So it actually, schizocarp breaks up into two carpels, and each carpel is termed a mericarp, right? So schizocarp breaks up into two mericarps, right? And what is a mericarp? It's actually a form of a carpel, right? Last one, nuts. It's in a situation where the entire fruit, not just the exocarp, right? The entire fruit is thick and hard, right? Now to the dehiscent fruits, the dry fruits, right? They crack open along seams and literally shed the seeds into the environment. So the seeds does not remain attached in this case, right? So one form is capsule, where the breakage of the seed happens along multiple sutures, and because of that, when the seeds open up, it look like a dry flower, like in this case. Follicle only breaks up along a single suture, right? I mean, you can see here and seen an example of milkweed. Legume is an example, another type where the breaking apart happens along two sutures, right? And as a result, the seed splits into two halves in that case, right? In contrast, in the case of milkweed, you actually see a single opening and the rest of the part of the seed coat remains intact. And then two more very similar parts, sleek and silicle. Silics are more elongated, thin and slender and long. Silics are much more shorter, right? There are other forms of dehiscence, but I'm only going to limit my explanation for these very basic forms. Now, I do want to go through this really interesting specialized fruits. I want to use the term over here compound fruits, because in all of those cases, it's actually aggregation of fruits. Many fruits aggregate, or many fruits come together and bind to become an actual collective fruit. One type is aggregate. Raspberry, thimbleberry, those are actually examples for aggregate, composed of many individual fused fruits. So in that case, several ovaries actually join in a given flower, right, to make up a single fruit that we eat. Then you have the multiple type, which is composed of many individual flowers fusing together to make a fruit. The example is mulberry, right? So here, different flowers, different ovaries in each flower, but when they mature, you only get a single fruit, right? So accessory, the last type, strawberry, is an example. So it's the part other than the ovary itself become fleshy, right? In strawberry as well as in blackberry, right? The receptacle, right? Remember the sort of the stalk of the flower expands and become fleshy and become edible, right? So because in strawberries and blackberries, it is the receptacle, it is not the ovarian wall, but a non-ovarian component that actually becomes fleshy. Botanically, blackberries and strawberries are actually vegetables. Okay, guys, that's basically it regarding the introduction lecture. We are going to go into the actual lab activity, right? Now, guys, I would really appreciate if you could, to become more interactive, open your videos so that I feel extremely less awkward. So if you could do that, I would greatly appreciate it. right? Any questions, guys, regarding your the lectures on the introductory part? It was a little longer, right? But I do appreciate you guys holding on.
Are we going to have to know the difference between monocots and dicots for the exam?
So the differences, we will actually go through some of those differences down the exercise part. So the some differences you already know since you asked are actually the root system. Monocots retain, dicots retain the taproot. Monocots do not retain the taproot, right? They actually, lose the taproot and becomes fibrous roots. And thank you for guys for opening up your videos. And another thing you will realize is actually the plant parts, right? How many in a flower, right? How many parts do you see in monocots versus dicots, right? Odd versus even parts, that's another difference. But when you, as we navigate through the plant exercise, you will learn other examples. So the answer is yes, we will cover many of those as we finish up the angiosperm lecture, right? And also while you do the homework too, you might actually have to look certain things up. Anything else you want to ask? Anyone else? Okie dokie, let's actually continue on with the lab exercise, right? So the first part is actually going through the key. I will do one of this, right? And let's take a look at the first example right over here. Look at this particular plant, right? And look at the flower. You see a single flower, and you do see many part in that particular flower, but it's a single, like sort of a consolidated flower, right? Let's look at the key. Try to break it up and understand what it is. 1A, 1B. 1A, flowers born in an inflorescence. B, flowers born singly. Please, guys, interact with me. What is it for the first image? 2 or
1B.
3? Emily?
I'll go to 3.
You will go to 3, flowers born singly. Of course, it's not an inflorescence. Inflorescence is actually when you have multiple tiny flowers in a single large structure. Yeah, we'll go to 3. Flower parts in 4s and 5s. Flower not white. Flower parts is in trees. Flowers white. Leaves with netted venation, right? Well, we'll go. What do you guys think? Is it 4 or is it trillium?
Go to 4.
That's correct, Matt. We'll go to 4, right? And the 4 is actually flower petals free, flower petals fused. Well, obviously, what do you think?
B.
That's correct. Petals are fused. Let's go to 5, right? Let's go back over here. Yeah, it's fused. You do not see individual sepals, petals. Flower petals 5, stamens many, not fused together, carpels many, not fused. Flower petals 5. Sorry. What am I doing? 4B. Flower petals fused. Flower is funnel form and blue. Yeah, it's Hypomia tricolor, right? That's the correct name, right? Yeah, you don't have to go to 5. It's B. 4B. Yeah. Make sense? Do we want to do another example? Yeah, we are good. Well, I have thumbs up, right? A thumbs up for do another example or good. We are good?
Can do one more.
Okay, I'll do one more. Of course, I will do one more. Let's do this one, right? So there's a hint. Each of these is an inflorescence and not just one flower, right? So you know immediately it's an inflorescence, right? So in that case, can you see like these tiny things over here? That's the actual individual flowers. This particular structure is the inflorescence. Okay. Number 1, A or B?
A.
A, correct. It's an inflorescence. Go to 2. Flowers born in a head inflorescence with ray florets and disc-like ray florets and disc-like florets, right? B, flowers born in a racheme inflorescence, alternating up to flower producing stems. So what is the difference between head versus raceme is actually in a raceme, you actually have sort of flowers producing in a, like this is a raceme, right, or a raceme, right, where you actually have a stem and multiple flowers coming out of it, right? And then so what is it? Is it actually going to be A or B?
A.
A, obviously A, right? So we go to, well, coreopsis, right? So yeah, so like that, it's actually pretty easy to understand. Let's actually go to the next exercise. Is actually understanding the life cycle. I will go over the life cycle and we'll go over smaller parts in the subsequent exercise activities, right? So let's start with the flower, right? Flower, in this flower actually has, both male and female reproductive systems. Now remember, there are certain types of angiosperms that produce separate male flowers and separate female flowers. In fact, sometimes you have both male and female flowers on the same plant. Sometimes you actually have male and female plants in different trees, right? And sometimes male and female flowers appear in different times. Okay, so remember that, right? We mentioned similar stuff in gymnosperms. The same applies to angiosperms. Okay, now anthers, let's look at an anther, right? The male reproductive system. You actually have the microsporocytes over there, and they undergo meiosis inside the microsporangium, producing microspores that are haploid. That actually has, a microspore has two types of cells: tube cell producing the tube, generative cell producing the actual pollen grain, producing the actual sperm, right? Now, the microspore develops into pollen grains. They actually exit the flower and or exit the anther and land on the female reproductive system on the top of stigma, right? And when that happens, right, the pollen grain, the tube cell of the pollen grain will develop the pollen tube, and then that will actually have two sperms, right? That is coming out of the generative cell. And the pollen tube will continue to grow, ultimately reaching the ovary and ultimately penetrating the ovarian wall and going and delivering both sperms into an ovule, right? Delivering the both sperm into an ovary, right? And then what do we have over here? Here we are looking at the ovary, the whole structure, and the single ovary is going to actually have multiple ovules, right? And then what happens inside those ovules? If you look at a single ovule inside the ovary, right? Again, ovary is this whole structure. Ovule is what you have here, right? There are 6 ovules in this particular example. So these ovaries undergo meiosis, right, and produce in the megasporangium, it produce single surviving megaspore, right? And then of course, that megaspore actually develops into female gametophyte, right? Inside the female gametophyte, you actually see different types of cells. On the center, you have central cells, right? On one side, you get these antipodal cells. On another side, towards the micropyle, you actually get the egg, the only egg, and 2 synoid cells, right? And well, and still you do see, this pollen tube, right? And the 2 sperms getting closer to the ovule. Ultimately, the tube will release both sperms into the ovule, and one sperm will fertilize the egg. The other sperm will fertilize or fuse with both central cells, right? Because of that, right, the central cell fertilization subsequently becomes the endosperm. It had 3 nuclei, so it is 3N, triploid. Now the egg is haploid, sperm is haploid. Their fertilization becomes the zygote, and zygote develops into the embryo. So this is actually how double fertilization happens. In this case, double fertilization continues. And then of course, zygote develops into the embryo. The central cell develops into the endosperm. I do also want to mention a couple of things regarding the central cell, right? Central cell is a single cell with two nuclei. Now, tell me, how on earth can that happen? Well, I'm going to ask you a question so that you figure the answer yourself, right? So what's happening in here, right, when you develop this central cells, egg, synagrids, antipodal cells, like how do you get those from the megaspore? Because the megaspore, as it matures into the female gametophytes, it's undergo mitosis, right? So, you know, in any cell division, right, whether it's mitosis or meiosis, you have two parts happening. One is actually the nucleus division after DNA replication, right? And the other part is actually cytokinesis, which is the cellular division, where the protoplasm separates around the nuclei, right? Here's a question for you. Should cytokinesis always happen after nuclear division? Again, let me repeat the question. Listen carefully, right? Should cytokinesis, right, the protoplasm division, always follow immediately after nuclear division?
Yes.
You would think so, right? Because that's how you were taught how meiosis and mitosis happens. Actually, this is an exception. Remember, there is always an exception to the rule in biology. In this case, you can ask, wait a minute, in that case, if you know, following Emily's point, which is actually true for 90% of the time, well, 99% of the time, cytokinesis happens immediately after cell division. This is the 1% where it doesn't, right? So when you create central cell, you only have one single central cell with two nuclei inside of it, right? So what happens is nuclear division happens, cytokinesis does not happen. That's why you don't have two central cells. You have one central cell with two nuclei, right? And that's how actually you have, upon fertilization, you know, you actually have this central cell containing three sets of chromosomes, two from the central cell, originally coming from the maternal side, and one parental um, nuclei coming from the sperm, right? And they all three fuse together, right, to make a triploid nucleus, right? Now, certain lilies, this is something completely extraneous, certain lilies undergo formation of a in the central cell, they first actually have two nuclei, and then those two nuclei fuse again to make a diploid single central cell nuclei, right? And that happens in only a few, like a very few plants. And lilies are the one that comes into my mind as an example. Not all lilies, some lilies actually undergo that, right? But generally, the general picture, two nuclei in a single central cell, right? And that becomes fertilized by one sperm. That becomes the endosperm, which provides the nourishment for the developing embryo, right? And zygote develops into an embryo, right? Now, the 3N triploidity is unique to angiosperms. The actual sustained dual fertilization is unique to angiosperms. Remember, again, gymnosperms have had two fertilizations, but only one of them develops. Here, both of them develops. And yeah, of course, the seed development continues. Ultimately, the embryo becomes a seedling, right? And then, of course, that seedling becomes a mature plant producing flowers, complete the life cycle, right? Now, remember, from the fertilization point onwards, these particular parts are not just diploid. It is a combination of both diploid and haploid, right? You know, until endosperm completely disintegrates, right? The seed will contain some parts that are triploid, right? That's actually something really, really important to understand, right? So here, actually, I kind of zoom into what happens inside the female gametophyte, inside the megasporangium or inside the ovary, right? You actually have first inside here, right? Inside the inside a single ovule of an ovary, you actually have this embryo, the mother cell, right? The ovarian mother cell or the embryonic mother cell. It undergoes meiosis, producing 4 haploid cells, right? 3 of those haploid cells die, right? Remember, always meiosis produces 4 daughter cells out of a single mother cell, right? And then, of course, and the surviving cell actually, undergo many form of modifications and ultimately, producing this 8 nuclei, particular 8 nuclei stage, through repeated mitotic divisions, right? And remember, when that happens, right, inside the central cell, like you have 2 nuclei, and those nuclei are called polar nuclei, right? And then the rest of the other nuclei, becomes either antipodal, and then 2 of them becomes synagrid cells, and only 1 of them become an egg cell, right? An egg cell lies right next to the micropyle, like this particular opening.
I have
Yeah.
a question.
Yeah, shoot.
What are the antipodal cells?
Yeah, you know what? Like, what do they do? Like, what are their function? Why do, like, we actually don't know what exactly they do. I actually cannot give you a straight answer, right? Like what kind of a vestigial? That's my only explanation I have. Like, they actually sort of a vestigial. They have a vestigial function, and frankly, I haven't read about cellular level plant reproduction. But I would be able to get back to you on that. I do want to do a little bit more research, and you don't have to know that, Emily. But right as of now, do they contribute significantly, substantially towards the reproductive cycle that you should be aware of? No, right? But I will promise I will actually get back to all of you on what antipodal cells do, what synagrid cells do. So any supportive function, none that actually I can be definitely sure of. Any other questions of the life cycle? Right? I mean, those are good questions. Why undergoing so much repeated, divisions? Why can't you just produce 4 haploid cells, right? And then 1 of them become the egg. Why 3 disintegrates, right? Why can't 1 of them become, why cannot 2 of them become the polar nuclei, right? Why not? Right? Those are good questions to ask, right? I mean, those are big mysteries in plant biology. So this is sort of, like the parts that you are going to learn in detail as part of the life cycle, right? And this contains the terminology that you are supposed to learn, that you are supposed to include into your lab notebook, right? Now let's talk about the zygote or the development of the plant body, right? So you first start with the seed. The first part that you see in any given seed, if you look at a bean seed, the part that you see over here, right, is actually called the hypocotyl, right, and the radicle, right? So it's actually what first emerges from a seed is the radicle. Radicle is the embryonic root, right? And on the top of the radicle is actually the hypocotyl, which is a part of the stem. Now, hypocotyl is a part of the stem. Radicle is what becomes the root. As the seed development progresses, right, the radicle develops the taproot, and then the secondary or the lateral roots emerges from that. The very top part that that was the hypocotyl sort of curves up, right? Sort of curves up, and that curved part keeps growing against the gravity, right? So the curves, the hypocotyl keeps getting longer. When it gets longer, what you actually interestingly see is a curve, like a U-shaped thing, right? And that U-shaped part surfaces from the soil. So when you first see a seedling developing, the very first thing you might see is actually the hypocotyl, right? And then or concurrently with the hypocotyl or after the hypocotyl, you will see this part, a part of the actual seed. And then what happens is the hypocotyl continues to grow, right? And sort of pulls the seed away from the soil and erects it upright like this. The bigger part of the seed you see are cotyledon. Those are actually the first leaves. Those are actually the first leaves, right? And cotyledon initially is actually green, and then it, turns brown or withers away, right? And then the actual first leaves, the foliage leaves emerge within the cotyledon, right? And they are actually green and remain green until they die. Cotyledons, why are they so large? Because they actually have absorbed nutrients and become taken over the role of the endosperm, right? Endosperm shrinks, and cotyledons, which are 2N diploid, becomes the source of nourishment for the developing seed, developing bean in this case. Now, at this point, you can clearly see the epicotyl is the part of the stem that occurs from the cotyledon towards the apical bud, right? The parts from cotyledon towards the root is called hypocotyl, right? So those are the two parts of the stem. Epicotyl, epi on the top, bottom part, hypocotyl, right? So remember the hypocotyl that you first see over here, that sort of emerging from the surface, right? That part is a part that underlies the cotyledon, right? So the epicotyl you do not see in these stages, because epicotyl is actually sort of embedded in between the cotyledons in here, right? And when the cotyledons give away, when they shrink, when they wither away, only then you will see the epicotyl emerging, right? All right, so again, stem has two parts: epicotyl above cotyledons, hypocotyl below cotyledons. Moving on.
Quick question, by the way.
Yeah.
Go
Oh,
ahead.
sorry. In a mature plant, is there any difference between, like, after the cotyledon, or I think that's how it was pronounced? In a mature plant, is there any difference between the epicotyl and the hypercotyl after it withers away and dies?
That's a great question. Once the cotyledons wither and die, no, because you cannot. I mean, like in that case, you need to figure out like where exactly this particular node is, right? Right. If you do, you can actually tell. But in a real mature plant, you will not be able to, right? To see epicotyl and cotyledon separate, epicotyl and hypocotyl separately. Like once cotyledon disappear, once they wither off, once they fell, you can't. Once they fall off, you cannot. Yeah, you all see just the stem.
Got it. Thank you.
Yeah, of course. So, I mean, I'm not going to go over this myself because, the parts I explained you should be able to tell the parts of a seed. Only part I want to point, this fuzzy part you see over here are actually the root hair, right? That is something you may not be able to easily tell. And then remember over here, you don't have to say like, don't say this entire part is stem. You can tell the two parts of the stem, right? The parts that is actually above cotyledons, epicotyl, below hypocotyl, right? And again, these parts are actually the leaf, the foliage leaf, right? And the cotyledons are actually the first leaves, which are rather than performing photosynthesis, they are actually storage in function. But I must mention that some cotyledons do have the ability to photosynthesize, right? Now, this is a dicot, this is a monocot. Now, I'm going to go back to one of Emily's questions. What are the differences between those two? Well, in dicot, you don't see a large endosperm. Endosperm actually tends to recede, right? Cotyledons take over the job of the endosperm in dicot. There are two cotyledons in a dicot. In a monocot, you don't see 2 cotyledons. You still see the endosperm, right? Not slithering or withering away, right? So those are actually 2 additional differences between the monocots and dicots, right? And these, you can see these, the first leaf emerging, right? And those are the foliage leaves, right? And then you see, and all the other structures you can label yourself, right? And then we will actually talk about the sporophyte. Remember, like the tree that you see outside, the seedling, the sapling, right? What is that? That is a sporophyte. Okay, where is the gametophyte? Well, if you actually see a fruit on a tree, yeah, the gametophyte is inside that, right? So now the labeling for a newly germinating dicot seedling is also going to be pretty much the same, right? So I'm not going to go over the labeling because, like the previous labeling I did helped you to identify what these parts are, unless you guys want me to let you answer these questions. Are you good? Yep, seems like I get nods. So one more point, like most monocots, the taproot on this cone seedling is still present. Remember, don't say that the taproot is absent in monocots. It's present. It eventually aborts, meaning it gives away, it disappears and become replaced by fibrous roots or by adventitious roots, right? So that's another difference between monocots and dicots, right? In this particular example, you don't see root hair because it's too young to develop root hair, right? But can monocots have root hair? Absolutely, they do. This one is too young to have root hair, right? Now remember, root hair are largely microscopic, but if you look at a seedling carefully, some of the root hair could be quite large, right? And why root hair are actually microscopic is actually a root hair is, the hair part is a single cell. It's not multiple cells. It's a single cell, right? Extending. So that's why actually it's largely microscopic. Alrighty, right? And remember the ploidy level, right? Do recall that most of the part of a germinating seed is diploid, but the endosperm is triploid, right? So I did label different parts of the shoot and the root. So the same thing applies over here. Only addition is I am showing you the fruit, right? And the flowers, right? And I did the labeling before. Now actually I am asking you to label all of this, right? Are you guys comfortable or do you want to do a little quiz over here?
I feel pretty comfortable.
Emily?
I wouldn't mind a quiz.
Okay, let's do a very quick quiz then. Number 1, what is this?
The leaf.
Correct. And this? More precise. Leaf is correct, but leaf is actually the entire part, right? This particular part only?
Is that pointing to a vein?
No, this entire part. Look at my pointer, not the arrow. What do you call that? Leaf blade, right? And this particular part, petiole, okay? This particular part, petiole, right? Now, Kayla's answer as leaf, that is not incorrect. I'm trying to be precise over here. What is this particular bud on the edge, on the end of the stem, on the top of the stem? What kind of a bud is that?
Is that the axillary bud?
The opposite, apical bud. And the bud you find over here is the axillary bud. Okay, now this part, this particular part where buds and petioles arise, what do you call that? Start with the letter N.
That's a node, right?
That's correct. The part in between 2 nodes,
Internode?
Correct. As simple as that, right? All right, so it's the same part over here. So this part I'm actually going to let you guys label yourself. It's just, always compare with my label diagram from the background, right? Now, just to help you better, I added two different label diagrams of flower structure. The structures are the same, right? The names are the same, right? But I just added a different picture over here. Now, what I want you to do, and again, the two critical parts that you want to understand that sometimes could be confusing is actually petals and sepals. Yes, in most plants, petals are colorful, sepals are green. Not always. Look at this monocot flower, right? The more colorful parts are actually the petal, right? The other parts are sepal, right? So in a given flower, if you have like two rings of petals, right? They are not two rings of petal, right? The innermost part that immediately surrounds the reproductive parts are the petal, and petals usually are more colorful than sepals, right? The outermost part is the sepal, right? Now, are petals always larger than sepals? No. So mostly, yes, petals are larger than the sepals. Are petals always more colorful than the sepals? Mostly, not always. Remember the sunflower? The sunflower sepals are actually larger and more colorful than the sunflower petals. So there are always exceptions, right? But when we ask you to label sepals and petals, you will not get a confusing picture. You will get a picture that you might have already seen, or where you can use the general rules to decide what is petals and sepals. Generally, petals are innermost, sepals are outermost. Petals surrounds the reproductive structures, sepals surround the petals. Right? Now, in monocots, do always petals and sepals look alike? Not always, but in monocot flowers, you will have a harder time telling them apart. But what is common between, what is unique to monocot flower is actually flower parts are in 3 or multiples of 3s, right? Over here, 6 sepals and 6 petals, right? I mean, some of the you can see actually 3 petals, 1, 2, 3, right? And then I want to say 6 sepals, right? So if you find flower parts like number of sepals, number of petals are either 3 or multiplications of 3s, you could be looking at a monocot. If the flower parts occur in 4s or 5s or multiples of 4s and 5s, you could be looking at dicots. Another difference between monocots and dicots, right? Now tell me, right? Let's do a short quiz. These colorful parts are what?
The petals.
Correct, right? And collectively, all the petals are called what? Think of a car.
Corolla.
Corolla, right? And what are these parts that are protecting the actual petals?
The sepals.
Sepals. And collectively, all sepals are what? Starts with the letters another C. Calyx, right? Again, this is something now I want you to label yourself, right? And look at over here. Let's do a couple of things. Organ that contain immature seeds, this particular part, what is that?
Ovary.
Correct. The immature seeds, or what will become seeds that are located inside the ovary, what are those? Ovules. Okay, those are ovules, right? And then this particular part, right, the male reproductive structure, the top part of the male reproductive structure, right, that actually produce pollens are called what? Starts with the letter A.
Anther.
Anther.
And correct, anther. And those pollens produced by the anther lands on what of the female reproductive system? Start with letter S.
Stigma.
Great, stigma. Excellent, right? So, well, again, the same parts I'm not going to go over. Seems like you guys are actually getting that very well. Let's talk about the seed now. This is a little important. Okay. Let's see. I don't know how far you have gone in your lecture, but I'm going to ask the question anyways. The entire structure is obviously called the fruit, and you can see the fruit has parts. You can see, you know, this fruit has divided into 2 parts, and each part is called what? Start with letter C. I said that's actually, you know, like collectively the part that contains
The
female.
carpel.
Sorry?
The carpel.
Correct. Carpel. Excellent, Randolph. Right? And then the central part is actually called placenta. I don't believe I mentioned that before. Now this part is called placenta. Again, the same nomenclature. There is no need to redo it. But remember, right, in previous one, right, in the tomato, you actually see sort of 2 carpels in this particular fruit. Here, I think I see 1, 2, 3 carpels, right? And now let's talk about spores and gametes, right? Actually, let me do something really quick over here. Yep. So in this particular diagram, let's label that couple. At least let's label some of them together. Before labeling, let me explain what we are looking at, right? Over here, the previous one, right over here, it was clear, right? A cross section of, in this case, chili, right? Or pepper. In this case, cross section of a tomato, right? So cross section of a fruit. Here we are looking at is a cross section of an anther, right? So this part is an anther. You cut across the anther, right? And you are going to see, you're sort of looking at microscopically the surface of this particular part, right? And instead of the entire part, you are sort of looking at just this part, right? You know, simply because you can see a bigger picture in that way. Now, I did not explain this part before, so I will label this for you, right? This entire tissue, right, is actually the anther, right? This entire tissue, right? The entire structure responsible for producing male spores, microspores, that is the anther, right? Which is the same as this particular part. What you have inside a given anther is 4 pollen sacs, right? And pollen sacs are actually diploid. Now, pollen sacs actually, after undergoing meiosis, they actually produce something called pollen tetrad. Tetrad means 4, right? Like you can see over here, 4 sets of cells. 4 sets of cells. All over you see 4 sets of cells, right? And here too, you see lots of 4 cells of threads. So inside the pollen sac, what you see are pollen tetrads, which actually are produced after meiosis, right? So pollen tetrads are haploid. Pollen sac itself is diploid, right? Remember that. And then this particular thing is actually this. That is the filament. Filament is the stalk that actually lifts the anther up in the flower, right? So this particular structure you see is the filament. Now imagine what's happened. This particular structure you see over here is the filament. Like how come that happens? Like imagine when you take the cross section, the filament is still inside the anther, right? So this, in this picture, this is the filament and you sort of saw it blown up over here, right? And then I said there are 4 pollen sacs. So you see part of a pollen sac over here. Another one could be like somewhere around here. You don't see all 4 in this particular picture, but there are. So let's take a cross section across a ovary, right? And so here, this is the entire female reproductive structure: pistil, stigma, ovary, and we are cutting across the ovary like this. And when you do, you see something that looks like this. So now, how should we explain this? Now, you can see this actually has divided. In this case, the ovary has divided into 1, 2, 3 large parts. Each one of those large divisions are considered locules, right? Now, I do not think that is in your terminology. I'm telling you that because it's out there in the literature, right? Now, each part within a locule, you actually get an ovule, right? The locule is another name for locule is actually could be carpel, right? Now, and inside a locule, you get in this picture, each locule has 2 ovules, right? And then this central part, right, collectively, right, this entire central part is considered the placenta, right? The discipent, don't worry about what that is. The discipent is actually sort of the rest of the tissue inside the ovule cross, inside the ovary cross section, right? That largely comprises the ovarian wall and sort of the filler between the ovarian wall and the locules, right? So what you need to know is actually ovule, you need to know, right? And yeah, that's actually something you do need to know. Locule, I just put it as an addition. Placenta, that also you do need to know, and I showed it before, right? And again, locule can be also considered as a carpel. Let's go on, right? So again, that's an ovule. That is the locule or the carpel, right? So based on that diagram, you should be easily able to label this. Remember, whatever that becomes the seed, like what becomes the seed? Starts with letter O.
Ovule.
Correct, ovule, right? And the entire structure, the overall structure is the ovary, right? And how many seeds do you think that you can get out of this particular ovary? You want to make a, anyone want to make a guess? Right?
6.
Yeah, that's correct. Right? How 6? Well, there are 3 carpels. Each carpel or each locule has 2 ovules. Each ovule has a single cell, right, that can go fertilized, right? So yeah, in that case, you have 6 ovules, so 6 seeds, of course, correct, right? Again, you know, this is actually sort of a blow up of this image labeled. This is for you to study and memorize. Well, we just went over what this, but over here, it's again, you know, like what I do is this is the same image as over here, right? Sort of blown up, and I'm kind of providing more guidance for you to understand what each and all of these structures are, right? The labeling is the same, right? So this is a good way for you to practice when you prepare for the exam. Now that's about the female reproductive part. Let's do the same for the male reproductive parts, right? Let's take a look at the anthers / the filament, right? So yeah, we were looking at the female reproductive part. We will continue to look at the female reproductive part over here, not the male, not entirely the male reproductive part. Still, we are looking at the female reproductive part, but instead of looking at the ovary, now let's look at the top structure, the stigma, right? So the top part is a stigma, and then you have the style, which actually a style connects to the ovary, right? So in a flower, again, what are we looking at? This is the female reproductive part, right? The pistil, stigma, top part, style, the tube-like part, which connects the stigma with the ovary, right? And you can start asking question, well, what is the point of the style? Why can't they get rid of the style and let the stigma be on the top of the ovary? Well, I believe it creates sort of a pollen competition, right? For the best pollen to reach, because growing the pollen tube, like you see in that particular diagram, and going through the style and actually reaching the ovary is a challenge. It creates sort of an evolutionary pressure for the best set of pollen or the best pollen to actually fertilize the egg or the egg cell, right? That's actually why there are very elaborate structures inside a flower. Now, if you look at this particular structure microscopically, it looks like this, right? In fact, if you look at more for, if you sort of blow up into this particular top part of the stigma, it looks like this, right? So many pollen grains can land on the stigma. So many, right? And a few of them can actually develop pollen tubes and start growing through the style, but only one of them will actually be able to fully fertilize, right? So in this particular diagram, if you were to label it, this is stigma. What you see over here outside with the pink staining is actually the pollen grains. And then this actual tube part that actually sort of lifts the stigma from the ovary is the style. Style actually has a sort of a tube opening inside, sort of a cavity inside. And what are these slight things growing inside the cavity? Those are actually pollen tubes coming out of the pollen grains, right? So now the labeling part, right? The top part of what is this? The top part of the female reproductive system.
Stigma.
Very good. And the tubes traveling down towards the ovules. What are those?
Pollen tube.
Pollen tubes. And the pollen tubes are traveling through what?
Is it called the style?
Correct. Very good. Excellent. Right. Now what we are going to do is, let me tell you what that is, right? We are sort of zooming in to one of these, right? What here is an ovule. We are looking at an ovary, a cross section of an ovary, right? And we are trying to look much more closer into a single ovule. And it looks like this, right? So when you zoom into an ovule, right, the entire structure is actually the ovary, right? And then an ovary has multiple ovules, and this particular part is the actual ovule. Remember, they both are diploid, right? And then the innermost part that actually has, those polar cells, the egg cell, that is the embryo sac. And remember, embryo sac has how many cells? How many nuclei? That's extremely important. 8 visible, countably visible nuclei, but only 7 cells. Remember, the polar cell, right, or the central cell actually has 2 nuclei in it, right? 2 polar nuclei. So remember that, right? 8 nuclei, but 7 cells, right? So this is actually more of a diagrammatic one for you to remember. Single egg cell, right? 2 polar nuclei inside the central cell. Okay. Now, once you understand that, you should be able to label this by yourself, right? So the confusion that students get is with respect to this part, right? So inside the angiosperm flower, right, you have the ovary. Inside the ovary, you have ovules. Inside a single ovule, right, that's what we are looking at. Inside a single ovule, you actually have the megagametophyte that is produced from megaspore, right? And megaspore came from megagametocyte. Megagametocyte, meiosis, megaspore. Megaspore undergo repeated mitosis, producing megagametophyte, right? And then that megagametophyte actually can be also called, particularly the megagametophyte of the angiosperm, can be also termed embryo sac. If you wondered what the hell is this embryo sac, it's another name for the megagametophyte of the angiosperm, right? Especially the one that actually has these 8 nuclei. Okay, so megagametophyte, right, that actually is in the process of developing 8 nuclei, right? If you go a few slides back, remember, come on. Which was that? Cannot remember which one that was now. Oh, in this life cycle, it's actually, don't know why, right? In this one, right, remember the megagametophyte undergoes multiple developments. Okay, so can you call this structure the embryo sac before you have the 8 nuclei? My answer is no. My instruction is no, right? You can call it developing megagametophyte, but when the megagametophyte actually produces the 8 haploid nuclei into 7 cells, now you can call that embryo sac. All right, let's go to where we were originally. Right. Okay. Anything like there's a lot we are covering today. Any questions? Everybody comfortable? Right. Okay. Please do interrupt me if you don't have any. If you don't understand something. Let's label this one, right? Well, immature seed, you know what that is, right? That is the ovule, right? Fertilized female cell, right? Remember, what do you call the fertilized female cell? Or the fertilized egg cell, what do you call that?
Zygote.
Correct, zygote, right? And the tube down which the sperm travels, what do you call that? Remember what happens over there, right? What makes that tube? The pollen grain makes that tube. The pollen grain makes it, made the tube. It actually, the pollen grain is still on the top of stigma, it sends something through the style, right? And then ultimately enters the ovule, enters through the micropyle, right? And that particular thing is still called pollen tube, right? It's only after entering the micropyle would it release the 2 sperms, for the fertilization to take place. So that the answer to that question, tube down which the sperm traveled, pollen tube, right? All right. Now I think we are actually getting towards the last few slides. Now this is going to be a little bit interactive, so I do want you to read with me and answer the questions. Rhododendron and ilex, right? Holly, right? Rhododendron, the English name is actually rhododendron. I was trying to look up a vernacular name. Some people call it mountain laurel, but what mountain laurel and is actually, mountain laurel and other forms of English names that you call this particular plant. Let me get my pointer. This particular plant belonged to the group rhododendron, right? Actually, I studied this, published a paper on this particular plant because it's really cool. It's a seed structure. The fruit structure is quite different between its southern and the northern distributions. It's very interesting. So this is rhododendron. Some people call it mountain laurel. I really don't like it because mountain laurel is a species, not a genus, right? I like this one. It's holly, right? And you all know what holly is, commonly known as American holly, right? Both have shallow root systems, right? They have thick leaves, and both of them are evergreen. They don't shed their leaves during winter. They can quickly take up water. They are both adapted to soils that are fairly acidic and shallow, meaning the roots are not that the soil isn't deep, right? The soil thickness is very shallow, right? You immediately hit the bedrock. And they are also adapted to live in lower light conditions, right? So now those are true features for these plants. Lower light conditions, younger, shallow soil, quickly uptake water, thick leaf. Where would you expect to find those? Thick leaf and evergreen. Where would you expect to find those? What kind of a habitat? What kind of a biome? What kind of a plant community? It's okay to be wrong, but this is really fun because I'm an ecologist. I love talking about adaptations.
Mountainous?
Well, because of the mountain laurel, you might think about that. Well, can you tell me, Emily, like that's not wrong, but I'm glad you mentioned that. Why would you consider a mountain habitat?
They have to take up water quickly and water runs down mountains.
That's not a bad answer because, like that, do you find them in higher altitudes? Actually, you do. Rhododendron, American holly, you find in lower elevations, but rhododendron in particular grow very well in mountain environments. Another reason is bedrock, right? I mean, the soils are much younger in mountains because, they undergo repeated erosion. So shallow soils are also characteristic to mountain environments. Good answer. What else? Where else would you find this kind of vegetation?
Would you find them more in like colder or very windier environments? Just because in that case they'd have to build up their leaves to be able to sustain the environment.
Correct. Colder environments. I mean, mountains are also colder. That's another reason why that answer was correct. Temperate environments, northern latitudes are relatively colder. They have a winter, right? But if you have thicker leaves, that thickness will prevent frost damage. You know what frost damage is? When the temperature drops below 0, right? If the leaf is not thick, the leaf tissue, the photosynthetic cells, parenchyma cells, can actually freeze. But if the leaf is actually thick, it protects sensitive photosynthetic tissues. So you're right, colder, cooler environments. Any other answers? Now think about the shade tolerance, the part about they can live in lower light conditions. Where would you expect these? In the canopy or in the understory?
The understory.
Yeah, correct. They actually are very prominent understory vegetation. You may think, wait, prof. American holly in my home garden, that's not in the understory. You are right. In your home garden, yes, but in the, if I take you a hike, right, you will find American holly as a tiny small bush growing under maple canopy, growing under oak canopy, right? Rhododendron largely grows in the riverine canopy, right? You might have very tall riverine trees like, sugar maple, beech, American beech, birk, and other riparian conifers. And these guys grow like a very dense scrub or a bush underneath very thick canopies. And also, both of them are very fire resistant. So they can actually grow in areas where there is fire. right? And actually, I'm big into fire-based land management. So let me give you a very short story behind this. Rhododendron, how many of you have seen rhododendron growing all over stream banks, particularly in mountain areas? How have you seen, observed that? Some of you are nodding. So do you have a reason, particularly if any of you actually have traveled in the southeast, the rhododendron growth around stream banks in mountain streams are extremely prominent, right? Historically, that was not the case. Rhododendron historically was not that prolifically growing, right? If you go way back to the history, pre-Indian time, right? Rhododendron was not a member of the understory, right? The reason actually it has taken over is that Indians try to burn vegetation so that they can hunt easily. And rhododendron is actually very fire tolerant and very disturbance tolerant. They actually they spread after fire was introduced to the landscape, right? So that's actually a really cool thing to talk about. There's a lot of stuff that actually now in my research, I learned about rhododendron in terms of their reproductive fitness. There are many strategies they try to enhance in the northern latitudes. In terms of reproductive fitness, because in the north, particularly in New England and Northeast, they are actually not that common. If you go to the Southeast, they are very common. So cool stuff. This one, right? Peace lily and the devil's ivy plants have leaves that have a thick waxy cuticle. Not the leaf itself is thick, the cuticle is actually thick and waxy. However, their leaves have a narrow tip. Look at this, look at over here, narrow tapering tip with a furrow for draining, right? I mean, if you look at the leaf itself, this midrib sort of creates a furrow. So it's not like a, let me actually show you to my camera, right? Leaf blade is not like this, it's more like this, okay? So that actually helps to drain water from the leaf blade. Also, even though they have a thick cuticle, their leaves are fairly thin, right? And very flexible and tend to be pretty dark green in color, right? I mean, you can see in this particular diagram, don't get fooled by the yellowness. The yellowness is because actually some of those plants, when we use it for horticulture, we hybridize them with other plants to make the leaf blade more prettier, right? Generally, naturally, they are very dark green. Question, what kind of habitats would you expect these sort of plants?
In very rainy, wet habitats.
Totally correct, right? Rainforests, humid forests, where you actually get rain. It could be tropical rainforests, it could be temperate rainforests, like in British Columbia, Oregon, Washington, So they actually are well adapted to rain, right? Wet, humid, dripping wet is what they're well adapted for, right? What and then again, the greenness, why do you think they're so dark green in color?
They get a lot of sun.
Yep. So if they intercept sun, that's correct. That's actually to maximize photosynthesis, right? To get the best out of the solar radiation. Where would you expect to find these kind of plants? They don't look like a tree, okay? They don't, and they are called peace ivy and devil's peace lily and devil's ivy, right? Well, of course, they are not trees. They are not. Don't even look like a shrub or a bush, but they are dependent on sunlight. Where would you find them then?
They grow on other trees.
Yes, they actually could be like a vine or like an epiphyte, right? So yes, they actually that's how they grow. That's kind of a habit thing. Good, very good, excellent job. This is actually extremely easy to tell, right? Carrion flower, jade plant, prickly pear, cactus are all examples of plants that have a very thick stem or leaves. In fact, what you might consider as a leaf is not a leaf. The leaf has become a spine. The stem has become something like a leaf, right? The green parts you see over here, right? This is actually, like those are actually, photosynthetic, right? So the actual leaf could be like a spine, right? So either the leaf has become a spine and the stem has become more succulent and green, right? But over here, this is actually, those are actual leaves. Stems are still succulent. What do you mean by succulent? Is actually fleshy, right? Adapted to store water, right? So the bottom, the opuntia, which is actually the prickly pear cactus, has modified leaves into spine. Like I just said, they actually have they have leaves, but those are like spines. They are not even green, right? They are not even photosynthetic. So as a spine, they provide more protection to the plant from herbivores than photosynthesizing, right? But in contrast, in jade plant and in carrion flower, have bitter organic compounds that serve the same purpose of spines, meaning that organic compound they produce in the carrion flower and in the jade plant, that organic compound is toxic and prevents predation, right? Prevents, rather, the predation for a plant is herbivory, right? It's anti-predatory strategy to protect the particular plant. All of them have very shallow roots. So rather than roots going deep, roots actually spread, right? That actually supports rapid water uptake, right? So what kind of habitat would you expect these plants? What would you be the point of spines and leaves? Well, we talked about all of that. Where would you expect those habitats? Well, what habitats?
Dry habitats.
Yeah, the driest possible habitat, like what? Deserts, right? So what would be the point of spiny leaves or bitter taste? Well, I just mentioned protection. So rather than saying protection, mention specifically what? Protection from herbivore, right? Sometimes the protection is protection from bad weather, right? So be specific when you say that. How many do we have? Nope. Just a few more slides to go through. Yeah. This one, again, like I love plants. I really wish we could have done this in person. I would make you guys more interested in plants than you are. Sundew, Venus flytraps, right? Two examples of plants that live in areas where water is very acidic, right? Water is not scarce, not water poor. Water is there, but water is extremely acidic. Soil is water saturated and acidic. Now keep that in your mind. Start thinking, what kind of habitats have water but very acidic? This type of habitats is limited in one of the macronutrients. Most of the time, these highly acidic habitats, because of the acidity, they don't have lots of critical nutrients like nitrogen, phosphorus, potassium. They're all the plants need for healthy growth. So because of that, some of those environments where you have acidic, waterlogged soil, nutrient poor, are very, less suitable for growth of certain plants. Yet, okay, but those plants you see grow in habitats like this, right? They have adaptations that allow them to supplement their nutritional requirements by being carnivoric, by catching and digesting insects, right? And sometimes, like sundew, they actually secrete a sticky substrate and the insects get trapped in that. Or actually they have modified leaves, like in the fly trap, right? Sundew actually, like here, if you can look closely, the edge of the leaf actually have this, glands that secretes a sticky substrate. So the insect gets trapped in there. In fly trap, these leaves are modified into a literal trap. Insects goes in and then the things actually shut down on the insect, right? And you guys know about this plant carnivory, fascinating plants. I couldn't, when I teach ecology, I cannot stop talking about them. So And so they actually trap insects to gather nutrients. Now remember, carnivorous plants do not absorb carbon from the insects they trap. They not steal like glycogen or glucose or proteins from them, right? What they do is they actually break down the protein and steal nutrients, inorganic matter, right? Carnivorous plants are not parasites. Carnivorous plants photosynthesize to make their own food, but for nourishment, they kill insects. Here's the question. What kind of habitats do you suspect these plants live?
Wetland environments.
Excellent answer, Emily. Can someone tell me, there are so many different wetlands environment. There are marshes, there are fens, there are floodplains, there are bogs, there are vernal pools. What kind of a wetland would you most likely to find these kind of vegetation with these kind of plants? Anyone want to make a guess?
Something barren, probably, or some sort of barrenness wetland, because if it has a lot of plants that die, then the decomposition would add to the soil nutrients, and their adaptation was specifically because there's no soil nutrients in the ground.
No, that's not a bad answer. I would not say you're completely wrong, but I would not use, I'm hesitant to use the word barren, because the environment where you find them are actually bogs, right? Peat bogs, peatlands, right? Acidic bogs. Those are the environments. Now, what kind of a plant, actually grows on those bogs primarily are sphagnum mosses. So that's why I said Darren was not completely right, because those are not barren, but mostly what you find on the peat substrate is going to be like one species, like moss, like sphagnum moss, right? Yes, some areas could be barren. For example, if a particular peatland undergo burning, and peatlands do burn frequently, regularly, not frequently, but predictably and regularly, you actually get these barren open wetland environments. The reason why I said, like, yeah, they are sort of barren, is because not many plants can establish in this extremely acidic, waterlogged, nutrient poor peat soil, right? There are only few plants that can establish. Now, can you guys tell me another plant that you actually grow in Massachusetts at industrial level, that grows really well in peat soil, that we eat, that actually the entire world knows? Like our state is historically known for this particular plant that we culture.
Cranberries.
Yes, cranberry, right? So there are only few plants that can tolerate this extremely nutrient poor environment. So cranberry is another one. But yeah. Now so yeah, the answer to the question is actually bogs, right? Wetlands is not wrong, right? There are, do I find these kind of plants in non-bog wetland environments? Yes, as long as you have waterlogged soil, poor in nutrient, yes, still that you get them. Like, I mean, there are certain fens, which actually type of a marsh, that actually have more peat in them, that still you get these kind of carnivorous plants. But bogs would be the best answer. Peatlands or bogs, right? What kind of macronutrients? Nitrogen, phosphorus, potassium. Out of all of those three, which one do you suspect to be the most limiting in peat soil?
The sand, maybe how like light it is. It's hard to have plants anchor in the substrate, I guess.
Right. I mean, your answer is what substrate, right? Sand. But I'm asking what specific nutrient, right? Between
Oh,
all
right,
the
right.
micronutrients. I mean, you're not wrong. You're not wrong to say
My bad.
sandy substrates are nutrient poor. You're damn right. They are nutrient poor, because it's hard for um, minerals to embed onto a sand particle. two sand particles actually don't have much water in between them, so nutrient drains away. So you are not wrong when you said that. But my question is, what macronutrients would be most limiting in peat soil? That's a little harder, but make a guess.
What about salt?
Yeah, what kind of salt specifically? What kind of salt? Salt is actually mini nutrients, right?
Yeah.
Yeah.
I'm guessing just like sea salt, like
Right, but sea salt has sodium and chloride, right?
right.
Right, like that. Now you know what I'm looking for. What particular element,
Oh, okay.
elementary form of the nutrient is missing or deficit on peat?
Hmm.
Did somebody already say nitrogen?
That's actually a correct answer, right? Nitrogen and phosphorus, right? So nitrogen and phosphorus, potassium are the three. Nitrogen, phosphorus, potassium, calcium, magnesium. Those are actually essential nutrients that you require in higher quantities, right? That's why we call them macronutrients, right? So even if you have them in lower quantities, plants are gonna suck. They actually are going to do really poorly. They might even die. They might even not grow well, right? So the answer is it's nitrogen and potassium for the most of the time, right? The reason is because it is waterlogged, it's harder for these soil particles to adsorb to nitrogen and potassium. And sometimes what happens, because it's acidic, what happens is acidic is what kind of mineral do you get in acids? H plus, right? And what is the form nitrogen exists in soil? Nitrates, phosphorus in phosphates, right? So sometimes hydrogen can actually sort of displace them, right? As like nitrate acids or phosphoric acids, and sort of remove those minerals from the soil, or make them less available for plants to absorb. So it's actually, the actual chemistry is a little bit complicated, but it has to do with acidity and the waterlogged condition. And then of course the microbial decomposition. Before microbial decomposition, what the hell is peat? Can someone tell me what that is?
It's like kind of moss.
Sort of. You are right. Peat is actually partially decomposed moss or partially decomposed vegetation. Why moss is correct? What kind of vegetation do you find in peat? Mostly moss, dead sphagnum moss to be precise, right? So what happens is, if vegetation undergo complete decomposition, you get nitrates, phosphates, minerals, blah 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 blah. You get all the inorganic nutrients, all the salts that James was talking about, right? But if decomposition is incomplete, like in the case of peat, it will not release as much nitrates or phosphates, right? So that's another reason why peat soil is nutrient poor, right? There's lots of nutrient trapped in the organic matter in the peat, right? Because it is waterlogged, right? Microbial activities will prevent, because of that, it prevents microbial activities. What microbial activity? I mean, decomposition is carried out by microbes. If it is waterlogged, it prevents microbial activities, right? Because microbes, for them to become fully active, they need to undergo aerobic, oxygen-based respiration, right? Without that, what happens is anaerobic respiration. Anaerobic respiration will not release minerals as much as aerobic respiration does. So for complete decomposition of vegetation, you need aerobic respiration and microbial activities, right? With waterlogged, you don't have enough oxygen for complete decomposition. That's another reason why bog soil or bog substrates are so nutrient poor, right? Good. So the last part is the root. We don't cover the stem. We only cover the root over here, right? In simply for the sake of simplicity. So what we do see over here is, like in this particular picture on the right side, it's actually an actual slide. On the left, it's diagrammatic. So outside you see the root hair, right? And then you actually see the epidermis around, and then inside you get the cortex, and then you can clearly see the xylem phloem, the vascular tissue, occur as a single circular formation, right? And xylem sort of occurs as a cross, right? And the phloem as sort of, in between the xylem, radii, right? And you can clearly see that the xylem is larger, has a larger cavity, has a larger wall. Phloem, smaller cells, right? And they actually, phloem are live, xylem are dead, right? So here we actually provide you a labeled diagram, right? Of a monocot, and here you are looking at a dicot. So Emily, this is another difference between dicots and monocots. The arrangement of the vascular bundle is quite different, right? Between between dicot and monocot. In monocot, you actually see xylem and phloem as multiple separate bundles that surrounds the pith, right? And over here, you actually see xylem and phloem together making a single centric bundle, right? So the arrangement is quite different between xylem and phloem, right, in these cases. Yeah, that's actually basically it, right? Again, here actually I provide side by side label diagram, and I expect you to answer which one it really is, right? So again, all the time, large cells with a thicker wall, xylem. Smaller cells with a thinner wall with smaller bundles, phloem, right? And outermost layer, epidermis. It is actually some epidermal cells that actually produce root hair. Well, any questions before we go to the homework, guys? Very well, I don't see any questions. Let's put the homework up here now and go through that, right? So the very first part in the homework is complete the lab, complete the table using your notes from your life cycle exercise and observations. If you have not done so already, record the information in your notebook. Monocot versus dicot. Emily, this is actually, your question at the very beginning. For example is corn for monocot, bean for dicot, right? And what you're going to do is actually, provide the scientific name, look them up, right? And then the phylum, again, look up the phylum, right? And I have actually probably have given the phylum example for eudicot. Oh, yeah, right. And also remember, like for monocots and dicots, they both belong to the same phylum, right? So if you look up one of them, I gave the phylum name already, so you have to only look up them once, right? Can someone remember what the phylum name is? What is the only angiosperm phylum? Start with letter A. Anthophyta, okay? Right, that's the only phylum you have to know. At least 3 differences between monocots and dicots, right? So I went over lots of differences in my previous lecture, right? What happens to the root histology, the arrangement of the xylem and phloem, the number of cotyledons, what happens to the endosperm, my bad, and branching, right? Branching versus lack of branching. There are so many things you can actually mention in terms of that.
So do we list like each difference as
Pair?
yeah.
Yep, correct. That's the best way to do it. Whenever I ask for differences, right? Tell, like, root in monocots is like this, roots in dicots is like that, right? Cotyledons of the monocot, this, cotyledons of the dicot, that. So make it a comparable pair, right? Differences. Yeah, I think there should be about, I don't know, 6, but we only need 3 here. Question 2, describe the path the sperm takes during the double fertilization from pollination until fertilization. Now think about the starting point. It says the path that the sperm takes, right? So first start with the pollen landing on the stigma, right? Pollen grain landing on the stigma of the pistil, right? And then remember the pollen tube grows, right? And then the sperm travels through that particular pollen tube. Then you actually have the generative cell and the sperm, and you have the generative cell that actually producing the actual sperm, right? 2 sperms actually, and that they keep migrating down the pollen tube, goes through the micropyle, enters the ovule, right? And then that's actually from pollen to the ovule. Like inside the ovule, what happens is actually the pollen tube enters the ovule through the micropyle, and then one sperm, both sperms are discharged, right, and into the embryo sac, right, and one fertilizes the egg cell, makes the zygote, other fertilizes the polar nuclei of the central cell and makes up the endosperm. That's actually basically your answer. Make sure to separate what happens in the ovule, what happens outside the ovule, right? And then compare the following, the flower. Complete the flowers. Complete flowers are comprised of, I cannot read today. Complete flowers are comprised of 4 concentric rings or organs, right? Of organs. So imagine this as a flower, right? This particular diagram, right? What do you find in the center, right? Think about, I said, like outermost part is what? Start with the letter S. Which is what? Sepals. And then petals, right? And then actually, like of male and female reproductive parts, like right in the center. No, it's actually the male parts next, female parts in the center, right? So the carpels in the center, sepals the outermost. That's kind of the generic plan. Are there variations to this? Of course, there are variations, right? But generic flower looks like that, right? And then let's look at and then complete that particular question. What are the reproductive parts? What are the the male and the female parts, right? Actually, so ovary, style, stigma, right? Filament, anther. Those are male. Those are reproductive parts, or you can collectively call them stamen and carpels, right? And everything else is sterile, right? So the receptacle, which you don't see here, the sepal and petal, they are going to be your sterile part. You are walking along and you stumble upon a small herbaceous plant. Although you don't know it yet, it's a new species, okay? Or rather, you take it to the lab, you key it out, and then you couldn't key it out, so it turns out to be a new species. Has never been scientifically described before, and you are responsible for describing it. Some of the characters you observe appear to be this, right? Radial symmetry. Flower has a radial symmetry, right? And flowers born in clusters. So you're looking at a compound flower, right? Or an inflorescence. Sepals green and fused together. So you have fused sepals, right? Not separate sepals, right? And petals 5. Right? Color is pink, purple, white, yada, 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 fused together into a single tube. So the corona, you have a fused corona, right? And 5 stamens fused into, fused with the petals, and 3 stigma, right? And you can see the number of plant parts, so is it a dicot or a monocot, right? Remember what I said, 3 and multiples of 3 versus multiples of 4, right? Like, so where would you put those in, right? You will have to do some research. And then the fruit is a capsule. We talked about different part of fruits, right? And remember, capsule is the one where the fruit ruptures on multiple sutures, right? classify your plant into a group, right? Is it like, it's anthophytes, right? What kind of an anthophyte is this? Monocot, eudicot, magnolid? In that case, you probably want to research all the form of groups within Anthophyta. What are all the possible forms of flowering plants, and what group would you put them under? Why? Now, remember, when you make these questions, you might actually argue that it could belong to two groups. Pick one group that you can best support, right? Now that you know it's a newly discovered species, name it. Well, when biologists name an organism, they often name it after the location, the physical characteristics of the discoverer. So let me tell you about a cool facts. This is our last lab, right? So a couple of cool facts. I actually describe new species. I unfortunately don't do it for plants. I do it for animals. I have named about 10 species so far. So yeah, I mean, basically the genus name is usually not name, right? You already have a genus, right? But sometimes the genus is also name, right? And the species, the species epithet is the one that most often is going to be new. I have never described a new genus, but I have described a lot of new species. So how do you describe it? Well, yeah, the location name, right? Right. So how do you Latinize the location name, right? If it is from Taunton, right, I could go Tauntonensis or Tauntone, right? So like that, you can actually Latinize the location name. And there are protocols how to Latinize, but that's actually beyond the scope of the class. Person's name, right? Let's say, like, I mean, you want to name it after, I don't know, like, your mentors, right? I actually have named a species after people that actually I respect in the discipline, right? Or historical characters that belong to that particular location, right? For example, there is a particular species of fish that named after Darwinensis, right? There are species named after a prominent evolutionary biologist, Richard Dawkinensis, right? And I cannot, yeah, there are so many ways you can actually name it after a person, or the characteristic of that particular flower, right? Or that particular plant, right? Spotted maculatum, right? Long, longinosus, or like that, you can actually, make the name characteristics of that particular animal, right? So the new common name, again, Darwin's flower, right? It could be reflective of the scientific name, right? So spotted flower, right? A flower with petals with a lot of, like that, you can actually provide the scientific name after any of those features, and then you can make your English name of the common name reflective of the scientific name, reflective of the characteristics, right? You may ask, I've been asked this name. So, oh, you have described this many species. Have you named all of them after yourself? What do you guys think? Do scientists name things after themselves? Do you think I name the species I discover after, like, Surasinghes? What do you guys think? Many of you are shaking their head. Say, yeah, no, scientists don't do that. I mean, it shows your ego if you do that, and we try not to be, right? We have people that we respect. Like, I have only seen, like, Linnaeus named things after himself, right? And there are very few species that have been named after the actual scientist, right? But other than that, there are actually, species that are actually, there's no example that that I cannot think of. Mostly we don't name after ourselves, right? Here's a fun question. In United States, right, there are, how do I ask this question? Does the name always go to a scientist? No, right? In US, let me ask you a question. What person who's not a scientist have actually most species named after? In the US, what person for animals, right? What person, a US citizen, that actually behalf of whom most animals are named after and who's not a scientist? Can I give you a hint? He's a president.
Washington?
No, you would think so, right? But no. I would, yeah, I was surprised when I learned this.
Roosevelt?
No, that would have been also made sensible because you know, Teddy Roosevelt actually have done a lot of work for conservation. Probably next time I discover a species, it should be that Roosevelty or Washingtony.
Was it Jackson?
No, no, not Andrew Jackson.
Kennedy.
No. I can give you a hint, but that's going to make things too easy. Fairly recent, I'll tell you that.
Bush.
No, but Bush actually has at least a species named after them. Don't ask me which Bush. I don't remember. I want to say it is HW, but I think at least one. Actually, Barack Obama, 9 species. Yeah, if you wonder why, it's because like actually during his time, he increased in the research funding twice, like by millions. So that's why actually he has so many species named after him. Most of the new species described based on NSF funding that was actually boosted from his stimulation were named after him. I was actually sure. I expected it to be, my bet would be Teddy Roosevelt because of his contribution to wildlife conservation, which is like a really big guy, you know, like we don't respect him enough. But yeah, that was actually a fun story. That's it, guys. We have actually we have 144, 134. We have a few minutes left. If you have any questions, I'll stay behind. If not, I will. Next time I'm going to see you is actually with the posters. Questions?
I've been really struggling with coming up with a genus and species name.
So here I'll give you a hint. I mean, think about it, because I based on the description, let me see what was the name of the location, physical characteristics.
Just in general for these assignments.
Yeah, I know it is a bit more like, I mean, I'm not going to worry about how good you do it. I mean, you've been doing well with like everything else, right? So the point assignment is 12 points, so that's quite a bit. Think about this. I'll give you a couple of hints, right? Radially symmetrical flower, right? So radioflora, right? Radio meaning radial, flora means flower, right? Right. Clusters, right? Flowers in clusters. So inflorescence. So inflorescences, right? And then what else? I don't remember what you call for purple. You may want to look up the Latin name. Hey, James, you took Latin. What do you call for, do you remember like what is the Latin name for purple or pink? Oh, for white?
Oh,
for multicolor? Like I cannot remember. I have some, I'm not as good as you for Latin. I have never taken the class. God damn it.
I really can't tell you, honestly. I don't even think we ever learned colors.
You probably might not have. I mean, that's something about you do, Emily. Like look up Latin name for purple, Latin name for pink, right? And then like sort of, you know, like use that.
How do you range it into a genus and species name?
Yeah, I mean, if it is a species name, usually like the species epithet, it goes by like, you know, inflorescences. That would be a species name, right? For a genus name, let's actually, you don't have the cis component, right? Or the nay component, for example, if I want to call it like, another name would be like, radii, RADEAE, right? That would be another, like a species epithet to say radial is radiating, right? So that would be two ways, like cis part, is actually how do you mostly see species names, right? But like how accurate you are in terms of following the exact international code for zoological nomenclature, I'm not going to go that deep, right? Any general sensible nomenclature that is actually descriptive of, the species characteristics is good enough for me, right? And the explanation you can actually mention, like, I mean, you can be creative in terms of the explanation, right? So we did not, did we mention where we found it?
No.
No. So you can mention, since this species was discovered in this particular town, right? Or in a bog, bogensis, right? Hydrangeas growing in water, right? Yeah, like that, you can actually sort of be creative, since, like what we haven't mentioned is where you found it, right? And in terms of like you asked me about genus names, okay, look at the characters I have given. Are there any genus that actually have pink, purple, white flowers with 5 petals that are fused together? Are there?
Look it up.
Yeah, you can look it up, right? What genuses which actually have that? Ipomia, I will tell you, Ipomia genus actually has those characteristics, all of them, right? So look up what kind of flowers actually have 5 petals with that kind of a color, right? What genus has fused petals? What genus actually? What are the genus that produce capsule fruits? Capsule fruits with 3 carpels, with 3 stigmas, right? Google those terms. Are those genus or genera? Can you think about a genera that actually, fit into those categories? Not all of them. Don't look for things that actually have exactly 5 petals, exactly pink, purple, and white in color, exactly 5 stamens, exactly 3 stigma, exactly 3 carpels. Like, no, not all A through F. Maybe just one. Pick one character, right? Look up the genus, right? Ipomia radii, Ipomia inflorescences. Like that would be the ways I would do it, right?
And what does
Now,
it mean by the stamens are fused to petals?
meaning actually, sometimes you can actually, the stamen, you can remove it, like completely remove the stamen from the flower receptacle. Like generally it's directly attached to the receptacle, the flower stalk, but sometimes it's attached to the petals instead, right? It could be, sometimes it's actually attached throughout the filament, right? Or throughout the stalk like part, sometimes like partly, right? It could be any of the ways. So meaning it's not connected to the bottom, it's fused, lengthwise it's fused to the petal. That's actually how I would do that. I know it's a little challenging, there's some creativity. That's a fun, like that kind of creativity is part of science.
And
Yeah.
what is a magnolid?
Magnolid is actually a magnolia group lineage that we did not go over today, right? I mean, because of simplicity, we don't go that group. It's actually another part, like monocot, dicot. It's sort of a basal group, right? An early evolutionary group within Anthophyta. Look it up, actually. That's an interesting group. Maybe I should have, I see, like I'm running out of time. I wanted to talk about that, but I let it go because there is not a whole lot about it. If you guys want, actually, I can put like a couple of information about magnolids, but if you ask, I will do it. Because Emily, what did you ask earlier for? Why there are like so many different cells? Like if you want something like that, I can actually look it up and upload information to the Blackboard. But
I would be interested.
oh, I love to. Well, that's actually any other questions? Good questions, Emily. Great. I'm going to let you guys go and I will see you. I will not see you in the practical, but I will see you on the 29th.
Can we still email you if you have any questions?
Email me, set up a meeting time, any of those.
Alrighty, thank you.
Of course. Bye-bye.
Thank you.
very, very much. Have a great
Yep.
day.
Bye.
You
Thank you.
guys too.
Have a good one.
Kenny, you have questions?
No, I don't have any questions.
Yeah, what did you say yes or no? I didn't hear what you said.
I said no. I just wanted to let you know that I uploaded my homework really late. So not really late. It was late, but you said that you don't accept late homework. But I didn't know. I kind of forgot about the homework, so that's why
I'll take a look. Since you uploaded it, I will respect your attempt and I will take a look at that. Yes, I mean, I did mention that in the very first day, like the, like you need to submit them. But since you have uploaded, I'll take a look.
Okay, thank you so much.
Yeah, absolutely. Anything else?
No, that was it.
Alrighty, I'm going to end the session. I'll catch you later. Bye-bye.
Thank you.